I'm Dr. Amy Turner and welcome to Protein Tech's podcast, the platform where we talk to life science leaders and get to grips with the big questions of the day that help us to read the book of life. I'm joined today by Dr. Julie Aspden, Associate Professor at the University of Leeds. She is a groundbreaking researcher in the area of RNA biology and specialises in translation and gene expression regulation, which it's fair to say is a crucial part of a lot of biology. Beyond her research, she is passionate about championing diversity in STEM and paving the way for women in leadership roles. In this episode, we are going to explore the growing world of RNA and highlight how increasing diversity and inclusion in research is necessary for addressing systemic biases in the sciences and technology. So welcome.
Thanks.
Just jumping in with kind of a very broad question. How did you get to where you are now?
So that's it. I had a really fun time doing my PhD and it really kind of got excited by how genes work. And I had a really encouraging supervisor and that just got me really excited. And from that stage on, I sort of decided I wanted to do a postdoc. And I went to the US to do that, which was really fun and a new experience. And then I came back kind of full of ideas and new technologies. I'd learned all about sequencing and fruit flies and came back fully equipped to do some even more fun science when I got back to the UK. And then I worked at the University of Sussex when I came back and learned some more skills. And then in 2015, I started my own group, which was like a dream come true, really. Like a kid, I was like a kid in a candy store, like having my own equipment in my own lab. So that's that. And then I've been running my own group ever since, doing research on ribosomes and translation, but also doing lots of teaching at the University of Leeds.
Amazing. So going back to your PhD, what was your PhD specialising in?
It was on the really detailed mechanics of how the ribosome, which is the machine in our cells that makes protein, how that works. And they were really very kind of precise, detailed experiments that weren't very exciting or glamorous at the time. But actually, some of those experiments are now some of the things that have become quite trendy that 20 years later, because actually what I was working on is some of the modifications that helped us, the field, develop mRNA vaccines and some of the things that went into that. We were doing in a kind of slightly different way back then. And so it's kind of exciting to look back now that something that didn't seem so cutting edge at the time now is really important and people, it's kind of helped contribute to that mRNA vaccine development. So
amazing.
Yeah.
And then your postdoc, that was in the US, you said. What did you specialise in there?
I was looking at fruit flies. That's where I caught the bug of using fruit flies as a model organism.
Caught the bug, I like that pun.
And we were working on another aspect of gene expression. So I was looking at a step before proteins are made and how the RNAs are created and how that's regulated. Thinking about it in different tissues in the fly, but also it sort of makes sense in terms of humans, because though it seems kind of slightly frightening to think about, but actually the way genes work and how they're expressed is very similar between a fruit fly and a human. Mm-hmm. So what we can learn a lot from them. And whilst I was a postdoc at Berkeley, all these new sequencing technologies became much more widely available. And I was really lucky to kind of be part of that revolution. And the people who were doing all the computational aspects were at Berkeley. And so I learned from them all this really exciting stuff. And so I was really kind of immersed in what we call the transcriptomics field then. So I didn't realize how lucky I was. So when I came back, I had all this expertise that was really useful. And that sort of, I've taken some of those aspects from my time as a PhD student, as a postdoc in Berkeley, and some from Sussex. And kind of, I used all those experiences to sort of create a niche for myself at Leeds. And that's how I sort of worked out what I was going to do in my own lab.
Nice.
When I got to choose.
Yeah, kid in a candy store, that does sound quite fun. So kind of going back to all the way back, what was the first thing that sparked your interest in science? Why did you choose this path that you've taken?
So I was really fortunate to have some really inspiring chemistry teachers when I was at high school. And one of them had a PhD in biochemistry. And she was not just a really smart person, but she was a really fun person and she made science super fun. And I think that was the first inkling that actually there was a pathway to do that sort of career. And then then I applied to do biochemistry as an undergraduate, and I really loved that. And during the summers, I did 2 projects at different universities. I had a summer in Cornell in the States where we spent the summer crossing maize plants in the field, because obviously you can only do that in the summer when you're working with a plant. So we did a lot of crossing the maize and then eating the maize after. Oh, wow. not the same maize that we genetically mutated, but that was a really fun experience. And then the summer after that, I went to the lab of molecular biology in Cambridge, and I had a fantastic summer there, being immersed in RNA and gene expression and ribosomes. And that was such a stimulating place. And actually, the time when I was there, that summer, is when the first structure of the ribosome, this machine that makes protein, was discovered and characterized. So that sort of lit another little fire inside me. But there also one experience I had actually before that. When I was a teenager, I did some work experience. It was just for a couple of days, actually, with this amazing woman she was a professor of genetics, but she was also an ordained priest.
Oh, wow.
And her job was basically, part of her job was to do postmortems on naturally aborted fetuses and try and work out why a baby might die, what might the issues be, and try and work out if there was a genetic reason.
Okay.
And then give parents genetic counselling if that was appropriate, to work out how they could move forward.
Wow.
So that was, as you can imagine, quite challenging emotionally, but also seeing that how genetics and science and understanding our genes had a really important impact on people's lives, and trying to understand mutations and when things go wrong and the impact of that. And I found it pretty inspiring watching a woman do this really challenging job, but also a woman of faith, and be able to kind of balance those two things was, that's a pretty challenging scenario.
Rare.
Yeah,
Quite
pretty
rare.
Yeah. So I think she was one of the people that really inspired me. So there's definitely a number of different people who's kind of working with them or watching them help me kind of work out the path I wanted to take. Yeah.
Nice. So at the moment, what does your group, in sort of a brief nutshell, what does your group actually do? What are you working to discover?
So we have two big questions that we work on in the lab. One is really centered on that machine that makes the proteins in the cells, and that's cells in our cells or bacteria or plants, or that same process is occurring in all cells. And that machine has about 80 different parts to it. And we always thought that they were the same 80 parts, never varied, and always did the same sort of job. But now we realize that actually there can be switches in and switches out. And there's a lot of diversity in this machine, the ribosome. Mm-hmm. And what's exciting about that diversity is some of it can impact which genes are then translated, which proteins are made, or how they're made and how it's regulated. So we're trying to unpick that conundrum at the moment. Mm-hmm. And we're taking a kind of fun approach. We're working in a variety of different situations, systems. So we're looking in viral infection, we're looking in plants, we're looking in fruit flies, we're looking in human stem cells, and we're looking in yeast.
Wow.
Yeah, I was slightly ambitious when I wrote this project, but thinking about actually what's in common. If we look across all these different types of organism, can we find some patterns? Mm-hmm. And does that help us kind of develop some rules of understanding this scenario? Because it really sort of changes the way we look at how genes are regulated. And it's by understanding this changes of the composition and how that translation, we can really impact a whole variety of areas. So we can, there are several diseases caused by mutations in these proteins. And so by understanding the ribosome better, we can potentially develop therapeutics and treatments for those patients. Viruses are really clever at adapting the ribosome to their own ends. And so if we can understand that, and we've understood it in a couple of situations now, we can start to develop therapeutics that take advantage of that little thing that virus does.
The Achilles heel.
Yeah, exactly, the Achilles heel. So that's one way it could be useful. Another way is actually in sort of more biotechnological kind of way. So if we understand how the regulation works, we can think, okay, so how can we manipulate the ribosome to maybe make more protein more efficiently when we're making a therapeutic?
Oh, interesting.
Or in a plant, then maybe we want the plant to be more resistant to drought or salt or something, so then we can kind of engineer the plant to do that more efficiently.
I've never really thought about it. I kind of knew what you did before, and I understood that you were kind of trying to answer these questions, trying to figure out how this engine worked, but kind of the broad impact that it's having is really quite remarkable. And I think the metaphor that was coming to mind when you were talking then was that the ribosome, in my head, it's like a car. And obviously you look on the street, you've got XYZ different cars. Every single one of them's got wheels, but they might have a slightly different, one might have a sunroof, one might be two canisters in the engine or whatever. My engineering is not very good, but you know what I mean? They're doing the same function, but in very different ways. And I think that's a really nice thing to kind of apply to biology. From my own background and from my biochemistry training, obviously the central dogma was always taught, right? So DNA to RNA to protein, that's how things function. And then obviously this idea that actually along those steps, there's different variations, and thinking about how that could impact expression regulation is really interesting. At what point did this kind of idea of specialized ribosomes, when was it born, and how many people are working on it at the moment? So
actually, it came up as an idea in the 1960s, 70s.
Oh, wow.
And then people sort of ignored it for a bit. And then there was some really pioneering work from Maria Barner's lab in Stanford in the States. And her group found some examples that illustrated how it could work, because it was more theoretical. And then when we could see these examples, then other people started looking in their own systems, and more examples were kind of elucidated. But they've all been in different species in different scenarios. So I think the thing that there's lots of people working on it at the moment from different perspectives. So there's lots of different types of variation. And so some people might be interested in one particular type of variation, for example, RNA modification, and then look at it in their specific system. So that might be zebrafish, or it might be human differentiation, or whatever it is. And so this idea of ours is trying to pull things together a bit, add to the knowledge, but also think, how can we understand some patterns?
Yeah, so kind of zoom out a little bit, get the big picture, and then zoom back in for individual questions that you want to answer.
Yeah, exactly.
Nice.
And it's funny what you were just saying, because actually the other side of the lab sort of challenging the central dogma in a different sort of way, because the other part of the lab is working on finding novel bits of the genome that we thought were sort of dark and dreary and not doing anything, we actually think are being translated to make little proteins that the field has ignored and thought, was just rubbish. So the dark matter of the genome, people said, that's not really doing anything. But the whole field is kind of developed, where people are starting to use some high throughput sequencing technologies and discovering new places that the ribosome translates. And then of course, we want to work out what these things are doing that the ribosome translates. Sometimes it's rubbish and it's noise, and it's actually part of the evolution of new genes, we think. So how a new gene might form. And then some points, that little protein might evolve a function. And so some of the ones we're looking at have functions, and some of them are like slowly edging towards a function maybe in the future.
So you say little proteins, have these got a name? Are they actually called little proteins?
Microproteins.
Okay, so it's kind of, obviously microRNAs exist and long non-coding RNAs. And I know a little bit about the kind of the world of RNA, but I didn't realize that there was microproteins as well. Is that a new?
Last 15 years, let's say, people have been finding these things. But scientists are really good at coming up with funny little names for things.
Yes.
I mean, a microprotein is defined as something that's smaller than 100 amino acids.
Okay.
And the reason, there was a fairly arbitrary way that when we started sequencing genomes and understanding genomes, a bunch of people clearly decided that 100 amino acids was a real protein and below 100 amino acids was not a real protein.
Mm-hmm. Okay.
So there was, like a lot of biology, it's a sort of arbitrary division, and then we realized, best guess that was wrong. And so I think this is one of the revelations, is that small can be really important. And some of these things they're different types of functions. So they might be regulating something big. Right. And they seem to have a role in kind of the immune system as well. But we're really we've got some super exciting examples. So our lab, we work on those that contribute to neuronal function and neuronal development. So we've discovered some that have really important functions in that process.
Okay.
And some of them seem to have a link with brain cancers.
Oh, wow.
So it's really important that we understand what these peptides are doing, because they might be doing something important. Yeah. Some of them might be just noise, but some of them are doing important things and associated with disease. I think one of the most important things I see is that they come from places from RNAs that people have characterized the function actually for the RNA having a function. And people have developed therapeutics for the RNA. And I think one of the most important things that we try and work out for not humanity seems a bit over, but
I think so. Humanity, let's go with it.
I think it's really important that we work out if the function of that gene or area of the genome is because of the RNA or the peptide or both.
Right.
Because if people start to develop therapeutics for one or the other and it's not that entity that's actually contributing the disease, then it's a bit of a waste of time.
What's your gut feeling?
I think there'll be times when it's the RNA, sometimes it'll be the protein, and sometimes it could be both. I'm sort of super excited about those examples. They'll be nightmarish to work on.
Yeah.
But I kind of like that kind of idea that there's sort of flexibility in the genome, that maybe it's on its way to doing two things at the same time.
I think that would be my gut feeling, is all these like feedback loops and all the more that you understand about the cell, the more that you think, oh, I actually don't know.
The less you actually
know. And it's quite interesting hearing you talk about the sort of level of complexity that you've seen possibly maybe in the last sort of 10 years. How do you feel like the field has changed in terms of the technology that we're getting, the amount of data that we get in? What's that look like over the last 10 years? Yeah,
I think a lot more labs are being able to afford large-scale omics experiments, which is great. That have more people having access to those kind of high throughput experiments is really good. And we're generating so much data. The worry is that we're quite often only extracting a small amount of it.
Right. Okay.
For that specific question we're asking, and we get information about that. But then there's all these other things that we could learn from that data set that I think across the fields we're not doing a great job of necessarily of, analysing it for different aspects.
Right.
But I think it's been really interesting watching new waves of these technologies come about. I remember when I was in Berkeley and there was a seminar, and the person who had invented this sequencing technology, which we now use to find our novel peptides, he presented how they'd discovered, and I was like, wow, this is super awesome. When I start my lab, I want to do this. And I was thinking, and we could do this adaptation and we can make it better. And I could, and that, in the end, that's what I was able, we could do that. And that was really fun. But now there's also different types of sequencing. So sequencing where you can sequence the RNA itself, not just a copy of the RNA in a DNA format.
Okay.
And so we're getting more and more information about RNAs, their length, their modifications. So I think one of the exciting things is to start thinking about all these different types of information we can learn at them per molecule and start to understand things, not just at a huge scale, but also huge scale, but on individual molecules, and start to work out more complicated things that up until recently you couldn't have even thought of trying to address or answer.
Well, this is the thing. I think up until recently, people have I mean, correct me if I'm wrong, quite often stayed in their lane. You know, you've got your biochemist, you've got your biophysicist, you've got your pure biologist, molecular biologist. And I think over the last sort of 5 years, computational biology has made such an impact in the amount of data that we can get and how we can analyze that data. From talking to you now, the lab group that you're in and who you've worked with, the collaborations that you've had, you're so interdisciplinary. It sounds like you've got aspects of evolutionary biology in your group, computational. How important do you think that is in answering these questions?
I think it's really important because there are very few questions now that can be answered in one type of experiment. Mm. And that's the experience I had as a PhD student. I literally did one type of experiment for 3.5 years. It was a beautiful experiment, but really that was a lot of the same experiment. Yeah. But it limits you to the type of question you could ask. And now it doesn't exist. You don't get papers where they've just done one type of experiment. Yeah. And I think my experience in Berkeley really kind of sculpted the way I approach that kind of interdisciplinarity, because there were so many good examples of people doing that. In different, it wasn't all RNA biology, it was all sorts of things, but I could see the power of combining structural biology with the genetics in the fly and the genomics. And so that I could see, even though I wasn't capable of doing all those things myself, I could see the real power of combining them and asking those questions in vivo. So for me, like using a model like Drosophila is super fun because you get to think about it at the tissue level, and there's, rather than flat plates of cells, where we can do lots of molecular analysis, but in reality, that's not what biology is like. Yeah. So I like putting together those interdisciplinary things. I also love collaborating with people. Mm-hmm. And so quite often the teams of people I work with, they're people I really have fun doing science with. Yeah. And it's fun to kind of try and think of things to do together where somebody's an engineer and somebody likes ribosomes. Okay, what experiment can we do together? Yeah. So it's also, there's lots of, I think friendship is a really important part of science. And
that's something that you say that, and it's something that I've never heard anybody say, but it's such an important thing to do. I think quite often people look at academia as very much pure research. That's it. And that should be your sole focus, is, answering that question because that's what you want to do. But there's such a light and a creativity in research. I know when I was doing my PhD, I loved that aspect of designing experiments and thinking about the fun things I could do with it, going into work and going, nobody's ever done this before. And that kind of light that comes from that side of science, which is not often talked about. But again, the friendships as well, much like most other areas of work as well. So obviously you spoke about the diversity in your research collaborations, the disciplines that you're working with. But you're also obviously very involved in increasing diversity and inclusion on a more of a systemic level in academia as well. Would you mind talking a bit about how you're involved in that sort of thing? Yeah,
so I used to lead a kind of gender equality organisation that exists in most universities now, called Athena Swan. And it's all about kind of trying to understand where we are in terms of gender balance in science, and then trying to think of ways to help and support to make sure that we're giving equity, ideally, to women. But actually it's more broad now, thinking, there are lots of different groups of people who don't have the same access to a scientific career. And even if they do get access, then there's lots of hurdles for them. And it's something I've been very lucky. I've come from a privileged background, and so I've had access at Oxford and Cambridge to sort of networks of things that I totally took for granted. But now as a group leader, I see people who haven't had that experience, and so they don't know how to access that information. They might not have the confidence to access or ask those questions because they don't have someone, a mentor, or they've had no discussions in their friends or family that would give them indications of what would need to do in that situation, or they might not have the financial support. I think this is one thing that I think has really positively changed, that when we were PhD students, This sounds like a really silly thing, but I think it makes a big difference. The expectation that is if we were going to a conference, we would pay for it ourselves and then we would get reimbursed. Now, my mum or dad would give me a bit of money to help me do that if I needed to. And I'm fortunate from that point of view. But then other people, I wouldn't have been able to do that. And then they don't go to the conference. And so that's a really clear example that if you don't have that, if you have less economic security, then you get less opportunity to do those things that others will have the financial security, take that risk to go to Cornell for the summer and not worry about how much money I was going to get from it. But I was just focused on the experience of learning about maize and rice plants and having fun in the field. Now, for many people, that's not an option, and they're thinking about how I'm going to earn the most money in the summer to then get me through next term. And so you start to realize, you see more and more students, you start to realize the pressures on other people that you haven't had to experience. And so I try really hard to make sure that to minimize any barrier that I think is occurring, to try and really encourage those people and mentor people, or suggest other mentors where I think I will have been no use to that person, but maybe I know someone who would be a really helpful person for them to mentor. So I think it's something that I've learned later in my career as I've seen other people, going through more difficult things.
Yeah. Well, I think it's so important to do that. Obviously, you say that. I never even think about this again because I'm coming from a relatively privileged situation, and then you see people who don't have that kind of access. But I think people from a diverse background, like you said, race, gender, economics, in science, it's so important to have such a variety, because not only is it just important, just as a life thing, it's nice to see people want to see, themselves reflected back as role models, but also people from different backgrounds think about things in different ways. And you're not going to have just a one-track mind. Two people who look the same from the same background probably have a similar way of thinking, similar way of designing experiments, and similar ways of thinking how their research could impact society. But if you increase diversity, inclusion, you get all this sort of more patchwork, I guess, this like more textured setting of the scientific field. And I think it's so important to have people like you put in the effort to try and kind of restore that balance of diversity and inclusion.
Someone once gave me some really good advice. There's no point hiring more people like you. You've got you.
Exactly.
You want people who are going to come with different scientific backgrounds, all sorts of perspectives that might be expertise. So someone in my group has worked in IT, but that's really useful. She has such a really good understanding of computational things that I really struggle with. So that's exactly what I need, is someone coming from a different perspective.
I think as a student in that sense as well, or a postdoc, whatever level they are, if that was me and I'd come to a group where I'm like, oh, I've got this niche talent, the confidence that you're building in that person is amazing. And I think, it's true of all areas of life as well. Obviously, you need a diverse team. You need people who are disruptors in the field. You need people who are going to kind of just do their job, the nice, come to work, do your job. And then you're going to need people who are pushing other boundaries in different directions. So it's great to have examples of teams that are built in that kind of nice textured setting. There's stats as well, going back to the gender thing, about businesses that have female CEOs typically won't make as much money, but they're a lot less likely to go bankrupt, whereas male CEOs will make more money because typically male characteristics are more risk taking. They'll probably on average make a lot more money, but they're also a higher risk of going bankrupt. So having a team of both females and males, these tend to be the most successful because they have the drive of, I guess, the risk taking, but the sort of more sensible responsibility head of a female. So that's like one very small example, but it does translate. I guess another question, how are you implementing that into, I guess, an academic sense? Are you working with any, I know Athena Swan is something that you've worked with before. Could you maybe touch on the processes that have been put in place since working with that?
Yeah, I think it's about looking for Athena Swan specifically, but I think more broadly about different career stages. So I think in generally, my experience has been especially for gender, there's a big drop off in the gender balance between postdocs and group leaders.
Right.
So we lose a lot of women at that point. And it's thinking about trying to work out why that's the case. And it can be for good reasons and people are making a positive move, and that's great and that's the right thing for them. But if people are not making that, trying to make that jump, are there ways we can, because they're not being supported enough, then how can we put that support in place? Mm-hmm. So I think mentorship is a really important thing to do. I think I'm someone who believes in team mentorship. So I've had loads of different mentors for different things, and then I would go to different people to ask for advice on different topics. Yeah. Not because I don't trust anyone in there, but it's actually just you benefit from the widest experience possible. But I think at that point, when people are thinking about, shall I try and take the next step in my career and try and have my own group as a woman, if there's anything I can do to talk about how I did that, how it felt, how difficult it was, the reality of it, but how rewarding it is also. And any people can learn from that or decide not to do it or to do it for their own and that's fine. But if I can help do that, I really like to do that. But also there's obviously, as then you go through, even when you are an academic and you've got your own group, we still aren't getting that many female professors in some areas. And biological sciences is relatively good compared with some areas, but it's also just mentorship again and making sure people have that support. And
do you think we're heading in the right direction? Do you think in 50 years' time it's going to be pretty even at all stages? Will it naturally get there?
I hope so. You still run into situations where you think, oh my gosh, yeah, I forget that there's some people who don't seem to have moved on in the last 30 years. I experienced sexism for the first time when I was 12 in terms of someone, and it was really impacted, and I can still remember it. That's how much it impacted me. So I was a prefect at school, and I was head of school, both schools I went to. And there was a boy who came up to me when I was 12 and said I couldn't be a prefect or head of school because I was a girl. And I was just like, what? And that was the first time that I ever felt that somebody else thought there were barriers to women, because I'd never, my parents had been so supportive, and I was only 11. Yeah. But it was beginning to that sense of actually women sometimes are told they're bossy, not encouraged to be leaders. And then that's pushed, squashed out of us sometimes.
Do you think for you that lit a little fire in your
belly? Oh yeah, of course.
Said, no, I am going to do that. Yeah. I'm going to, you know what? I'm going to be a prefect. I'm going to be the head, and I'm going to, in the future, run my own group.
Well, I think I found, though I find leadership terrifying in equal measure, I've always enjoyed it. I always wanted to sort of step up and do that. And I was lucky that in my school situation, I had lots of experience of doing that. And that I think then helped at every stage, trying to get involved in things and leading things on a whole variety of kind of levels. So when I started at Leeds, with two of my colleagues, Neve and Mary, we started at very similar times, and we could see there was something missing in terms of the omics community at Leeds. And so we came up with this crazy idea. We pitched it to the deans. They gave us some money, and we started this virtual group, Leeds Omics. And that was it was really using our expertise and stepping up and leading and saying, this is what we think we should do, let's do it. We weren't being asked to do it, it wasn't part of our job description, and that sort of thing comes naturally to me, and I like doing it. I mean, that's not to say that when I'm doing it, I'm then terrified and I'm racked with lack of confidence. But
make it.
But I want to have the vision to do the things, and then I'm like, oh no, why am I doing this?
But it's got to be done. You're a change maker, I guess. Maybe you're the disruptor in the team. You're the person
I'm not sure about the disruptor. I'm too good at tissues.
The boundary pusher in the right direction. Let's call it that. So going back to Leeds Omics, what is that Maybe describe what that is.
So it's sort of virtual group of people at the University of Leeds. It's anyone who is interested in omics. So it might be proteomics, might be transcriptomics, metabolomics. There's a new omics every few months, I feel like. So it's anyone who's interested in doing that. And we come together for seminar series. We have training workshops. We have a coding club, which is really good because it's a really kind of encouraging space where we have our bioinformatician Elton, who picks themes once a month, and students and postdocs and even PIs and technicians, if they want, can come and have a discussion on a specific topic, like they might be struggling to code certain thing or trying to do an analysis. And so there's like Elton does a bit of training and did a discussion, and people sort of do some peer mentoring and helping. And that's really a good thing, I think, because then you've got people who might be studying cuttlefish, and there's people studying brain cancer, and then there's someone studying the structure of some membrane protein, and they've got similar coding issues, but they're coming together to work and think about it in terms of the omics. So it can be really useful and empowering, I think, and it's really fun to learn about people's biological question definitely. But actually the core of the kind of technology is overlapping, so it's sort of a different way to think of things. And there's lots of people across the university, medicine, all types, faculties who were interested. So it's
a really nice community way to kind of upskill yourself in a way that's really beneficial for multiple different research areas. Yeah. Amazing. So you said cuttlefish there, and it made me think you obviously work with flies, Drosophila. Why flies? That's my question.
That's a good question, because I was in a lab that was working in flies. In Berkeley, there's a really strong tradition of Drosophila research. Right. And I realized whilst I was there how powerful and useful it was. I did have some experience for a few days of working with mice and rats, and decided that was not for me. Yeah. And at some point I did think about switching to zebrafish, but I'm glad I didn't. Flies are a little easier to care for.
Are they, in terms of the work that you're doing, is there like are they a better model organism compared to a zebrafish?
Oh, this is now, we're going to have a very controversial.
Yeah, amazing.
Now, the zebrafish are good because they're vertebrates. Right. But there are fewer tools available. So there's the pluses and minuses. And you also have to keep them in water. Yeah. Like a fish. That's quite a lot of work. So yeah. But their embryos are see-through, and they're really good at sort of screening lots of little zebrafish and looking at the embryo and development. So they're a great model for doing that. Drosophila was also a great model. I think there's also C. elegans, which is a very popular model. I think C. elegans and fly people get really, really confrontational.
The worm versus the fly.
The worm fly.
Avengers assemble.
Yeah, totally. The one thing I always have to admit that C. elegans is great because you can put them in the freezer and then take them out like a year later and they're still alive. That's a very attractive quality in an organism. Yeah. We cannot do that with a fly. Yeah, you cannot do that in a fly. Yeah. But the flies have lots of other amazing attributes. They can fly. Yeah. Yeah. They're also, they only live for about 30 days. So you can do lots of experiments relatively quickly compared with for a mouse, for example. Obviously, the bits within the tissues within a fly are smaller. So one of the tissues we work on is the testis. And people are always like, oh my gosh, that must be minuscule. Yes, it is. It's very small. But, you do have those tissues. And there are actually, there's lots of developmental questions that you can work on in the fly that are really great. And there's lots of technologies and tools and things to make genetic tricks and mutants and things in the fly, which are great. But, it isn't a human. But there's lots of, as I said, lots of the gene expression.
I think you'd find it would be difficult to find a human whose testes you could work on.
Yeah, exactly.
In a lab. So I think for the greater good, it's best to work on a fly. Cool. Do you enjoy working with the flies?
Yeah, I mean, I think lots of people don't like the smell of.
That's what
it's not the smell of the fly though. It's the smell of the food.
Okay.
It's the kind of yeasty kind of food that they like to eat. I think it's that people don't like. But I think it smells like a sort of bread. Beer kind of.
Bread and beer, you can't go wrong.
Yeah,
really.
Exactly.
So I'm going to ask quite a broad question, but it might be fun to answer. What in your entire career, what do you think is the most unusual experiment? The most unusual experiment that you've ever run, and second question is, what's the sort of biggest breakthrough that you've ever had in your career?
So I remember when the first foray I had into doing cryo-electron microscopy, and we had this crazy idea, me and my collaborator. Could we take the ribosomes out of fly testes and then solve their structure by EM? But there's, they're very small.
Mm-hmm.
We're going to have to get lots of them. So we spent, poor Taya did all the hard work. And Michaela, we spent a long time dissecting flies, getting all the testes out. And then we thought, we're not going to have many ribosomes. We're going to have to really concentrate. We purified the ribosomes, concentrated. And then we sat there on the microscope and looked at this first set of ribosomes. And one was like, oh my God, I have never seen so many particles.
Wow.
Because we were so scared that we weren't going to have enough ribosomes that the whole grid was just full of them.
Amazing.
And then we had to dilute them. But that was kind of a really fun moment because we were like, I'd never seen EM visualization before. So it was really fun to see that, the ribosome. That was kind of fun to do.
Am I right in thinking that you were also instrumental in developing a new ribosomal purification method? The polyribo? Yeah,
so we do. It was a variation of a technique lots of other people had contributed to. We made a kind of slight improvement to it to ask our specific question about our novel ORF, the small ORF. So we sort of made an adaptation, and that was my idea.
So talk to us. What was the method, and why would you need it?
When we were looking for those novel translation events, what we wanted to be sure about is that it wasn't just one ribosome trying an RNA out going, well, should we try this one? Nah, let's go. So we wanted to be really sure there was at least 2 ribosomes engaged in translating. And so rather than looking at all the ribosomes and what everything was translating, we isolated polysomes first. And polysome is the term we used for multiple ribosomes. Mm-hmm. So then we basically isolated RNAs with at least 2 ribosomes on, but not too many ribosomes, because then that open reading frame wouldn't be making a micropeptide, and making a big thing.
Okay.
So we sort of size selected the small things, and the aim was to then be more confident that we were detecting real translation events.
And how do you do that? Just for a very lay audience, how would you actually, you've got, let's go from the top. We've got an organism, let's call it a human. Then you scale down, you've got the cells, or the organs first, then you've got the cells, and then you've got these little machines in the cells called ribosomes that translate your RNA into protein. How on earth do you get a ribosome out of a cell?
So it's actually relatively easy. I'm glad people think it's difficult, because it makes people think that we're really good and capable. But essentially, if you take cells, and there are 2 main compartments to the cell: the nucleus, where all the DNA is, and in the cytoplasm, that's where the ribosomes hang out and do the translation. So we essentially just pop the cells, spin the nuclei so we get rid of all the DNA stuff, and then we're left with the soup of the cytoplasm where the ribosomes are. And then we just put them through what we call a gradient. So it's basically a tube with increasing concentration of sugar in it. Mm-hmm. So they're having a very, very fun, tasty time, the ribosomes. And we spin that material through, and it separates out the complexes, the things based on the number of ribosomes. So if there's like 10 ribosomes bound to an RNA, it goes near the bottom of the tube. And if there's only one ribosome bound, it stays near the top.
Right.
And it's so beautiful. We can see when we look at the absorbance, UV absorbance down the tube, we can see these lovely peaks, and we can see, oh, that's got 2 ribosomes. Oh, that's got 3 ribosomes. And that's one of the things everyone in my lab loves. That's one of the things. Makes you a true Aspden group member if you get super excited about seeing those peaks that nobody else cares about. But that's a fun thing to do for the first time, because people are always like, wow, I can actually see my ribosome complex.
I mean, it is interesting because obviously you do this every day, right? You're so engrossed and very enthusiastic, which is lovely to see, about your research. But you know, from anybody listening or watching who's never done this before, it is, what you're doing is actually incredibly amazing. As scientists, I think you do get quite bogged down in your questions, your trade. You very much specialize, and you kind of forget how cool is it that, you know, you can see these things. We can drill down into our biology, flies biology, see these things that we can't see them to the naked eye. And it's really nice to chat to you and see how enthusiastic you are about the science, because it's quite infectious, really. And it's really great to see. I guess one question would be as well, have you encountered any challenges in your career? Obviously, you seem like you've, you know, climbed to success very quickly and in such a nice way. But was there any challenges in your career that you could talk about? Yeah,
definitely. When we made the move back, so I was with my husband and I both had postdocs in Berkeley, and we had a great experience. And we'd been there for 5 years, and we wanted to move back to the UK to be closer to our family. And he had a lectureship position, so he was sort of making the next stage up. And I just couldn't find a position. I applied for fellowships. I didn't have enough papers, so I didn't get any of the fellowships. I was applying for postdoc positions, and I couldn't get one, and I was getting quite down about it. And then in the end, I saw an advert for a technician's role that sounded really super interesting. So I applied, and they were like, you have a PhD. This is not the job for you. I said, but I really want, this project sounds really interesting. And so they've just made a job for me. They had enough money. So I joined, and I did what the I did that sort of project, and I brought all my expertise to that, and it was really fun. But that was a big challenge at the time, because I wanted to step up and be a group leader at that time, and it didn't work out. But then I got some really good experiences at Sussex. My mentor at the time, he taught me a lot about how to write a grant and focus on those papers. And so it was, in some ways, I wasn't ready for the next step. But then when after 3 years at Sussex, then I was really ready.
Yeah.
And then, you apply for lots of jobs and you don't get them. And I remember not getting the first, I think I had 3 or 4 interviews and I didn't get any of them. And I was getting interviews, so that was good. And then I had 2 of my mentors at Sussex. So I said, could you give me a practice interview? Because maybe I'm just doing something really wrong, and that I don't know that I'm doing wrong. And they gave me a practice interview. I gave them my talk, my spiel, and they're like, then they quizzed me with really difficult questions. I was like, oh no, this is terrible. And then afterwards they said, we have no idea why you haven't got a job. You will get one. And I was like, oh.
Pat on the back.
And that was really good, because it gave me a bit more confidence that I wasn't completely on the wrong track, and I should just keep going. And then the next interview I went to, I got the job. Maybe it was just that confidence boost.
Possibly. I mean, it's sliding doors as well. You could have got the first interview that you did and then ended up at a dead end and not done this amazing career that you've had.
Exactly. I wouldn't have ended up at Leeds.
Well,
and then I wouldn't have been doing EM or met you, or so I think it's good to think of it that way around. I think trying to get that position as a group leader is really, really tough and can be really depressing at times, because there's an encouragement to apply for fellowships, and that's a fantastic route, but it is really competitive. And if you don't have enough good quality papers, then it's really difficult. But I had somebody come up and say, look, your CV is not good enough to get a fellowship. Mm-hmm. So stop doing that. Here's a grant that I think you should apply for, that I think you have a really good chance of getting. I applied for that grant and I got it.
Right. So it goes back to, again, mentors and that kind of thing, using who's in your network. What would be your biggest piece of advice to, let's say, your younger self?
Focus more on the papers.
Yeah.
Also collaborations. I think working with other people makes you a better scientist because you learn from everybody. You can learn something from everybody. And I think that's helped a lot. You can get more papers that way, but also you make friends, you make colleagues, find mentors that way. So that's, but do something fun, right? Like, and the world changes, I think you have to be creative about, okay, well, this idea I haven't got funded, but there are other ideas out there. Let's think creatively. Maybe I'll come back to that. In 10 years' time, it might be the thing to get funded. There's funding is, you have to kind of ride the wave at that time and respond to what's going on. And sometimes your pet thing isn't what's going to get funded.
Yeah.
So I think you have to be a bit pragmatic and creative about it. And then that's also when collaborations are really good because you can start to influence the people around you. So now there's lots of people at Leeds far more excited about ribosomes than they were before I arrived. Yeah. And the novel orths, there's 2 virologists looking at novel orths in their systems. So I'm having fun working with them and learning from them, and they're learning from me. And so I think, make the most of those types of experience, I would say. Yeah.
Amazing. Just going back to what your answer was about the challenges in your career, you mentioned your husband is also an academic. How do you find sort of a work-life balance in academia? Maybe you could touch on your personal life as well.
Not very well. So one of our challenges is trying to find jobs in the same location. So we were lucky when we were at Berkeley. We worked very physically very close in the city, and then since then we've never been able to find jobs in the same city. So when we came back, we lived together and we'd been married for before we went to the States. So we've been married for a fair long time by this point, but then we ended up living apart so we could both not have to put up with terrible commutes because we found that was driving us a bit insane. And so then we were only seeing each other at the weekend. And then when I moved up to Leeds, he was still at Southampton and he was flying down, living in a hotel Monday to Thursday, flying back up. So none of those scenarios are conducive to family life. So, then when he got a job at Newcastle and I'm at Leeds, and so we live in the middle, and I have an hour's commute on the train, which I think lots of people say, how do you do it? And it's like, well, it is difficult, but we're living together. I have a fun job. We both have jobs, we're happy where we are. Ideally, it would be great to be at the same university, but it did mean that I was 40 when I gave birth to my child because it was quite difficult even consider having a kid when you're trying to work in the lab all hours God sends and not living in the same location as your partner. Limits the it also limits the time to create the baby. So I think that was a challenge, and I think I'm really lucky. My husband has just as much respect for my career as he has for his own, if more, if anything. So, when we had our little one, we did shared parental leave, which really, really helped me be able to manage my group and here's his group. So what we did was I took off we took some time off together right at the beginning, and then I had I think it was a 12-week block where I worked and he went back to work, and then he came home for 12 weeks and looked after the little one, and I went to work, and then we did 10 weeks, then he did 10 weeks, then I did 7 weeks, and he did 7 weeks, and that was really good because it made us joint parent, like we were really joint parenting, which I think is really difficult to do when generally the dad goes back to work after a few weeks, and then the mum ends up being the primary source of information and but that set the scene of that balance, and it also helped me run my group whilst I was on leave, and he's got PhD students and post, it worked for both of us. So it is definitely a challenge, but I'm lucky I have someone who like really wants to see me succeed and super proud when I have an invited talk. He's like, yeah, you have to go to Denmark. You have to go.
That's great.
And I'm like, that means 5 days with a little one. Oh, yeah, we'll cope.
Oh, amazing. I mean, it's also really great to see, obviously, you're a mother in science too. So obviously a woman in science is fantastic to be represented. But I know when I was doing my research, there was on a few occasions, women who were mums said to me, you kind of have to choose if you want to be really successful in academia, if you want to own your own group, that's your baby, or you choose a lower sort of position in academia, and then you can also have a family, and you have to sacrifice something. And I remember it on at least 2 occasions being told that at quite a formative time in my career. And it's I think at the time I sort of wanted to have a perspective and think, no, I could succeed in both, but there wasn't many examples of women who were succeeding in both. So it is great to have you as a person in my stratosphere that is shown to be doing that pretty well. And it kind of goes back to, I guess, your work in equality as well. I suppose, do you feel that because of your life experience, having a child later on in life, do you feel like that's also a bit of a driver for you to increase equality and accessibility within your current role?
Yeah, and think more creatively. So that was the solution that worked for us. But it's not a great solution because, the older you are, the more complicated pregnancy and having a kid can be. But I've seen that there's lots of examples out there of people who have had kids when they're PhD students, postdocs, technicians. I'm now seeing people come through who may have had kids, and then they've come back. And we've got a PhD student she's doing her PhD having had kids and coming back to that scenario. So that, and that's something I really want to support, because I think that is one model. There's no,
there's no one size fits all.
No, totally. And that's great. And I really want to make sure we encourage those people to have that option. Then, I think having kids when you're a PhD student must be really challenging, but people do it. And I have a colleague at work. So I think she had one child when she was a PhD student, one child when she was a postdoc, and she's doing incredibly well. And I go to all these people for advice, because their kids are older than I, and I'm like working out what to do. So I think there's challenges with each scenario, whether it's coming back later in life, having a kid as a PhD, having a postdoc, or being a PI when you're doing it. I think we should just try and make sure that we can support people to do whichever scenario they choose to do.
Definitely. And
try and make sure there's things to help support that. So it might be financial support. So make sure when people come back from parental leave that there's money to support them to do some extra training or go to that conference. They may not have anything to present, but going to that conference will then kind of immerse them back in that will then kickstart their next few years. It's those things where it's just, it might not be the priority for the group leader, but actually if we can insert some cash to help these scenarios, it will help that person's career and get back in having had time off. And that might be time off for caring for a parent. It could be having a child, or it could be sickness. But, those kickstarts, I think, make a big difference to people's career.
Definitely.
And it's not that much money in reality to do those sorts of scenarios.
Yeah, exactly. So obviously we've talked about your career up to now, and the wonderful things that you've been doing. But how do you think your next sort of 10 years are going to go? What are you aiming for? Big question.
Apart from world domination,
obviously.
I find it really difficult to think about what 10 years looks like, because things change so rapidly in science. I think one of the things that I'm obviously really excited about the projects we're doing at the moment. Yeah. One of the things or sort of next wave of type of thing I want to do is start to use this direct RNA sequencing technology, where we can learn, as I was saying, about the specific molecules themselves, how the composition of the RNA, the poly A tail, so that's the part on the 3' end that sort of contributes to stability, length of time the RNA hang around, modifications on it, and the specific splicing pattern. So you get lots of variation of which bits of RNA are kind of stitched together. And then linking that with its translation and how well that molecule is translated. And so it's sort of putting together all these features that we know something about, but trying to incorporate things like machine learning to see if we can make some predictions and say, okay, can we learn anything, some patterns about here? And then we can say, okay, we look at that RNA and we know what's going to be happening with it because it's got modification A, poly A tail length B, it's likely to, this is the scenario. So we could, that's what I would like to start doing.
a big network of, if I press that button, this is going to happen, and like filling in on all of the gaps of the RNA
sort
world.
of the lifetime of the RNA, the history of it, and how that impacts the end game. Yeah.
I suppose, like you mentioned, machine learning. That's an area of research or an application that's going to generate questions that we couldn't even dream of right now. So it would be really exciting to follow the research and see where it's going to take you, really.
Yeah, I think the people who've obviously learned these machine learning skills are going to be in huge demand, because I think it's only going to get bigger. I think we just need to work out the right questions to ask and how to generate the right sort of data to be able to then do the machine learning and choose the right type of approach.
Definitely.
So we need really good people to be able to do that.
I suppose we might have time for one more question about the broader impact of your research. Obviously, you're very excited about the nitty gritty details of these microproteins, these RNAs, how they're working in the cell. Is there sort of a broader impact to sort of on a society level, to a kind of a public audience in your research? Is there a kind of bridge there?
Yeah, I think this is one thing that during the COVID-19 pandemic, it was really clear that lots of people didn't understand how genes were expressed, and that the fact that there were being developed RNA vaccines, and that there wasn't really the education of people to be able to kind of understand the pros and cons of that and the implications of that, and that it wasn't going to suddenly mutate inside us. And it's that must have been frightening to people who didn't know what an RNA was. And I was like, well, yeah, I'm going to have an RNA vaccine. but I think it highlighted how important, firstly, that RNA research is. I already knew that, but it was good to have some. But also thinking about, actually, we have a responsibility, because if we expect people to want to take RNA vaccines, we need to explain what happens, why we've made them, what's going to happen to them in our bodies. So I think one of the things we're doing in our big project at the moment, where we have sort of a really fun capacity, because there's a big group of us, to do some more public engagement and type work, we're trying to do some sort of increased understanding of the ribosome itself and that process of decoding. So thinking about from our DNA to the RNA to making the protein, and what that process looks like and why it's important. Yeah. And so we're trying to do that. We've got some activities going to brownies and scouts and cubs, schools, and also public engagement events that happen in lots of. So we're one of the parts of the team is in Sheffield, and they collaborated with us with an artist to produce, and the artist produced a piece of work about the ribosome and this composition change. And so people were coming and looking at the piece of art installation, and hopefully, that educated a group of people a bit more about what the ribosome is.
Definitely.
So gosh. I think there's lots to be done. I think I find that difficult, that trying to explain things in a really simplified way that other people, that is accurate, but usable. And I think that's a challenge to people. And some people are really good at that. For me, it's difficult. But we've got some people in our team who are really good at it and really creative and thinking about how do we explain this scenario to people.
Yeah. I mean, you mentioned COVID before. I think that skill to translate science to all audiences has probably been made more apparent in the last 4 years, obviously, because people who had never I've never even heard the word RNA, had it being put into their bodies. And like you said, they were scared. Or this kind of education of all levels is something that I definitely have seen increase over the last 4 years. And I think it is so important. And I think also it kind of comes back in a nice cyclic way to diversity in science, right? You've got people who it will spark, going to an artist who's made this installation of a ribosome and thinking, oh, I was a really arty person. I thought I was going to go down like an art route and never even think about science. And now I'm going to think about science, and I can see these different possibilities of how I can employ my skills. And I think that's really, I guess trying to make the point of this cyclic thing of, communicating science, is very important for just education, but also increasing diversity and who's interested and who different ideas coming into it.
Totally. And I think that's a lot of going into a school setting, for example, or brownies or comps. I think that's a big part of it, because, and I feel a responsibility as a woman to be doing that more than I am, I think, because it does make a difference. So one of my little one's friends tells other people, and she's 5, and she's telling so-and-so's mum is a scientist. And, and that very statement of a 5-year-old saying that, and she tells lots of people, and they're probably not interested. But that clear, I think it's really important that we set that tone at that age group, that it is a pathway, and hopefully it should be open to everybody, whether that gender, race, economic background, anyone that is interested and wants to learn more about the world. That's a really exciting career pathway. Definitely. And it can take lots of formats. So I think just showing that it exists and how fun it can be, hopefully it will encourage the next generation of people, 5-year-olds.
Definitely. You're the reason why the classic kids drawing of a scientist is no longer a wacky white man with wacky hair and a lab coat. It could also be anybody. So thank you so much for this conversation. I've had such a lovely time talking to you, and hopefully the listeners of this podcast will have learned something. And I'm going to say it, if they want to get in touch for collaborations, then please do get in touch with Julie. If you enjoyed this podcast, this episode, and would like to learn more about the big questions in the life science research, then like and subscribe to our socials in the link below. Thank you very much.